This presentation is preceded by a video on initiation of DNA replication. Here, I am going to talk about DNA transcription in a bacterial system. I would encourage the interested audience to take a look at the previous videos as well. The material contained in this presentation has been taken mostly from the book Fundamentals of Molecular Structural Biology. We all know what is a gene. We also know that in the cell of a living organism, DNA is a genetic material. In other words, DNA carries the genetic information of the cell. This genetic information is transferred from DNA to another macromolecule, RNA, in a process called transcription. There are some similarities as well as differences between replication and transcription. Both involve the synthesis of a new strand of nucleic acid. However, in transcription, the new strand synthesized is by the addition of ribonucleotides and not deoxyribonucleotides. Like DNA synthesis, assembly of ribonucleotides in transcription is not random, but directed by a DNA template strand. Here again, DNA segments are unwound and bases unpaired, and nucleotide incorporation takes place in accordance with the principle of base complementarity. The other differences are that in replication, the entire genome is copied once and only once in every cell division, whereas transcription copies only specific segments of the genome and generates 1 to several thousand copies from a given segment. Further, unlike replication, in transcription, the RNA product does not remain base paired with the template. Hence, for transcription, there is a need for an enzymatic machinery that fulfills the following requirements. First, recognize transcribable regions of the genome and interact with the DNA duplex to make single strand available as template. Second, convey ribonucleotides opposite to the template bases in the active site in accordance with the principle of complementarity. Third, join two RNTPs by phosphodiester bond. Fourth, extend the RNA chain by continuous addition of RNTPs and then peel off the growing chain from the template. And finally, terminate at a specific site, that is, dissociate from the DNA. In all the three domains of life, bacteria, archaea, and eukaryotes, the key constituent of the enzymatic machinery is the multi-subunit RNA polymerase. 5 subunits in the common core are conserved in all the three domains. Here we have RNA polymerase from E. coli and that from yeast. If the two structures are superposed, similarity is observed between the two enzymes, particularly around the active center. Differences, however, exist in the peripheral and surface structures as the two enzymes have to interact with different sets of factors in the entire transcription process. Let us look at the core assembly of the bacterial RNA polymerase. 5 subunits join together sequentially. 2 alpha subunits form a dimer. To this dimer are added beta and beta prime subunits sequentially. And finally, the 5th subunit, omega, is added to form the core. The bacterial RNA polymerase is the simplest form in the family of RNA polymerases. Hence, it has been most widely used to study the phenomena and the mechanisms of transcription. Interestingly, the structures of RNA polymerases from the Thermus genus were determined much earlier than that from E. coli. However, a high degree of sequence conservation is found to exist among the RNA polymerases from all bacterial species. Its overall structure resembles a crab claw with 2 pincers forming a cleft that binds DNA and contains the active center. The largest subunit, beta prime, forms one pincer called the clamp, while the second largest subunit, beta, forms the other pincer. The bases of the pincer are joined to the N-terminal domains of alpha subunit dimer that serves as a platform for RNA polymerase assembly. Omega serves as a beta prime chaperone. If we look at the structure of the bacterial RNA polymerase core, we find that the cleft between beta and beta prime is partitioned into 3 channels. The main primary channel accommodates downstream double-stranded DNA and RNA-DNA hybrid. The secondary channel is for the entry of nucleotide triphosphates. This is another view of the secondary channel. And third, the RNA exit channel separates RNA-DNA hybrid strands. The active center is located on the back wall of the primary channel. It consists of a long alpha helical bridge helix connecting the beta and beta prime subunits. 2 flexible alpha helices of a trigger loop, an extended F loop, and a catalytic loop with 3 aspartates for coordinating essential magnesium ions. All these features have their respective roles in nucleotide addition. The core enzyme itself possesses the basic RNA polymerization activity, but it cannot specifically bind DNA sequence at the beginning of a gene to initiate transcription. It needs the association of a transcription factor called sigma. E. coli contains multiple sigma factors, but the most predominant one is sigma 70. Sigma 70 falls into 4 independent structural domains, as shown here. Each domain is further subdivided into subdomains. Besides, there are a couple of flexible linkers that allow significant movement to domains sigma 1.1 and sigma 4. For transcription initiation, the core polymerase binds the sigma factor. The sigma-bound form of the enzyme is called the RNA polymerase holoenzyme. There is an extensive interface between sigma 70 on one side and beta beta prime on the other. These are mostly made of salt bridges, hydrogen bonds, and van der Waals contacts. The specific site in the genome to which RNA polymerase holoenzyme binds to initiate transcription is known as the promoter. In a typical bacterial promoter, such as the one shown here, there are two conserved sequences, TTG-ACA and TAT-AAT, which are present respectively at positions -35 and -10, where +1 is the transcription start site. They are called the -35 and -10 regions or elements. Subsequently, a few other sequence elements were found to be present upstream or downstream of the -35 and -10 elements. As for example, there is an AT-rich up element upstream of the -35 and a GC-rich discriminator downstream of the -10. These elements also contribute to the specific recognition of the promoter by the holoenzyme. Components of the holoenzyme bind to these sequence elements of the promoter, as we shall see next. During promoter binding, alpha CTDs interact with the A tract of the up element via their helix-turn helix motifs. Side chains of an arginine and an asparagine are inserted into DNA minor groups, while two basic residues are involved in salt bridges with the DNA phosphate backbone. Alpha linkers are fully extended. Domain 4 of sigma binds the -35 element, while the domain 2 binds the -10 element. Part of domain 2 is inserted into a major groove. Domain 3 of sigma interacts with Tg, while a beta prime zipper interacts with -17 or -18 element. Now, bacterial transcription initiation consists of the following steps. First, association of the core enzyme with the specificity factor leading to the formation of the holoenzyme. Next, recognition and binding of the promoter DNA by the holoenzyme and formation of the closed promoter complex. We have already seen these two steps. What we are going to see next are the conformational changes, that is, isomerization in the closed promoter complex and formation of the open promoter complex, followed by conversion of the open promoter complex to initial transcription complex and formation of the first phosphodiester bond between RNTPs at +1 and +2 sites. This will lead to the synthesis of RNA of a critical length, and subsequently loss of the RNA polymerase promoter DNA contacts that will end up in gradual dissociation of sigma and escape of the core polymerase from the promoter. And finally, a highly stable protective ternary elongation complex will be formed. The closed promoter complex is intrinsically unstable. As already indicated, specific binding of the holoenzyme to the promoter DNA causes isomerization in both the molecules. And these are going to be shown next. Here, in the process of promoter melting, alpha CTDs and up elements which were in the bound form in the closed promoter complex are now disordered. On the other hand, there are interactions between aromatic residues in domain 2 of sigma and bases from -11 to -7 of the non-template DNA strand. As a result, an initiation bubble is created, and 13 base pair of promoter DNA from the -11 position to just beyond the transcription start site is in an open state. The open complex is stabilized by multiple interactions between the DNA and RNA polymerase amino acid residues. Contacts between domain 2 of sigma and the duplex form of the promoter, which were polar and van der Waals in the closed complex, are mostly replaced by aromatic interactions in the open complex. Here is an example. An adenine at the -11 position in the non-template strand flips out of the duplex, and this is accommodated in a pocket comprising 3 aromatic residues in addition to 2 polar residues. So, a thymine base at the -12 position in the non-template strand is still base paired, but no longer stacked with the -11 adenine. However, a tryptophan dyad, which is aromatic, forms a chair-like structure to interact with the -12 T. The first tryptophan of the dyad forms a pi stack with the face of -12 T. Next, a cluster of basic residues interacts with the phosphate backbone of the template strand from -13 to -10, bends it by 90 degrees and pulls it into the main channel. This action puts the DNA at the +1 position into the catalytic center. Now that a 12 to 15 nucleotide long transcription bubble is formed and the start site is in an appropriate position in the active center, the open promoter complex is converted to an initial transcription complex. The first phosphodiester bond is formed between the RNTPs positioned at the +1 and +2 sites, and RNA synthesis begins. There is no requirement for a primer. Two NTPs interact with a number of beta and beta prime residues in the active site. After the formation of the dinucleotide, the polymerase tends to move further downstream to continue with the transcription process. However, at this stage, the upstream DNA RNA polymerase contacts in the initiation complex are stable enough to hold back the polymerase. So, unable to move forward, the polymerase instead pulls the downstream DNA from +2 to +15 while unwinding it. This is the phenomena called DNA scrunching. In the process, DNA compaction stress builds up in the initiation complex. As the nascent RNA attempts to grow beyond 4 or 5 nucleotides, a conflicting situation arises in the initiation complex. The RNA encounters a steric hindrance by the sigma 3.2 loop. The tip of the sigma 3.2 contains a cluster of acidic residues. So, there is charge repulsion between this cluster and the 5' phosphate of the RNA. The charge repulsion and the stress associated with DNA scrunching forces the polymerase to synthesize short pieces of RNA and prevents it from escape from the promoter, that is, moving beyond the promoter vicinity. This is abortive initiation. Alternatively, and frequently, the nascent RNA, instead of falling off itself, may cause disorderliness in the sigma 3.2 tip and push its way towards the 4th domain of sigma. Once the polymerase is able to synthesize about 15 nucleotide RNA, the sigma is released. The polymerase escapes from the promoter and proceeds towards elongation. In an upcoming video, I am going to talk about elongation and termination of transcription.